Start again with our talks, and what I was asked to talk about was really a large project that has been funded and sponsored by the Caring for Carcinoid Foundation called the Caring for Carcinoid Neuroendocrine Tumor Biobank Consortium. And just to tell people what that is, the concept is in fact relatively simple and relatively straightforward. This is, at this point, really an agreement among 5 independent institutions, all of which are focused and have programs focusing on neuroendocrine tumor patients to collect clinical information, blood, and tumor specimens from patients who are consenting, who are willing to participate in this particular database in the consortium. One of the things that is often really tricky about using information that is collected at many different sites is that the information is collected in different ways and things are coded in different ways, and that very often makes it almost impossible to analyze data or analyze specimens across many different institutions. And as one can imagine, since this still is not the most common tumor, it can be quite critical if one wants to look at large numbers of patients to be able to do that. And that's exactly what this consortium is designed to do. To standardize not only the collection process, but also to standardize the database process so that samples and data can be analyzed. The participating institutions at this point, there are 5. There is Dana-Farber Cancer Institute right here at Mass General Hospital. Dan Chung will be leading the effort. At Memorial Sloan Kettering Cancer Center, Diane Reedy will be leading their effort there. At Stanford, it will be George Fisher, and at MD Anderson, it will be James Yao. So that is the Biobank Consortium. Now, I'm often asked to talk about databases and consortia, and I always sort of dread it because it's sometimes a somewhat difficult thing to get people excited about. And I thought what I would focus on in this particular talk is try to explain why these biobanks are so important and why they're becoming increasingly important as some of these new treatments are being developed. There are obviously many reasons that they're important, but one of those is that they can really help us identify promising new treatments. Additionally, they are really necessary these days to identify molecular predictors of treatment response, particularly in the age of these new targeted therapies. And finally, as we'll talk about a little bit here, and I think hear about later on from Matthew Meyerson, these really can be a resource for discovering new treatment targets. We had a question earlier, I think, from Ben about how long does it take to develop a new treatment for patients who have carcinoid or neuroendocrine tumors. And in fact, I have just a little story to illustrate exactly that type of effort. Several years ago now, I think about 5 years ago, we were actually quite fortunate at Dana-Farber to have support from, I think, Steve Kofer, who's here in the audience, as well as others, to start developing a biobank right at Dana-Farber. And at that time, there was already the beginning of interest in targeted therapies for neuroendocrine tumors, but of course, we really had no idea which therapies would be of interest, which therapies would work. So we did, at that time, a fairly straightforward experiment. These days, it might be considered even a little bit too simple. But we knew of certain targeted therapies. We knew what they targeted. So we took a tissue microarray, an array of tumor specimens, and simply looked to see if carcinoid tumors had those targets. So one of those targets is called c-KIT. This is a target for a drug called Gleevec that has been extraordinarily successful in certain types of sarcoma. Another of those targets was EGFR, the epidermal growth factor receptor. That is a target in lung cancer where drugs like Tarceva have been very successful. And then the other targets we looked at were called vascular endothelial growth factor and its receptor, vascular endothelial growth factor receptor. And lo and behold, just looking at it very simply, the target that was expressed most highly in carcinoid and pancreatic neuroendocrine tumors just happened to be VEGF and its receptor, VEGFR. So what we did at that point is thought about, well, what kind of drugs might target this particular receptor? And at the time, there were a few choices. One of those drugs is bevacizumab. It's also known as Avastin. Another drug was sunitinib, and another one was sorafenib. And in truth, we really could have chosen any of these. As it turned out, no one was terribly interested in carcinoid tumors at the time, but we were able to take that data and show it to what at the time was a small biotech company called Sugen, who made the drug sunitinib. And we're able to convince them that it might be of interest to do a trial of this drug in patients with neuroendocrine tumors. So we did that, and this was a phase 2 study that Jennifer Chan, I think, talked a little bit about earlier. We were basically just looking to see if you give patients who have neuroendocrine tumors this drug sunitinib, do the tumors shrink? Do they benefit from the treatment? And indeed, it looked like they did. These are results that came out just last year from this particular study. And if you look, all the patients on the right-hand side of these curves had at least some amount of tumor shrinkage, suggesting that sunitinib really did have activity in patients with neuroendocrine tumors. This really isn't enough to convince absolutely everyone that the drug is effective, and we were talking earlier again about the need to do a randomized study. And based on these results, a randomized study was in fact done, and those results became available only about a month ago. They're still in preliminary form, but really quite intriguing. This was a randomized study where 150 patients with pancreatic neuroendocrine tumors received either treatment with sunitinib or placebo, and it turned out that sunitinib more than doubled the time it took these tumors to grow. It more than doubled time to tumor progression. I think providing more data that this type of treatment really can be helpful for patients with neuroendocrine tumors. If you think about the normal course of drug development, it might take 10 or 20 years to develop a new drug and then show that it's effective and make it available to patients. This is, I think, a story where that whole process was made much, much faster, again, by the availability of biospecimens integrated both with scientists and the ability to do clinical trials. The process in this case was speeded up really to only a few years from an idea to something that has become, and I think will become, much more widely available for patients. So that's one story, one example of how biobanks, I think, can help. There are a number of other ways that biobanks can help improve treatment for patients. And another question that often comes up when patients come see us in clinic is, well, what's the chance that the treatment is going to work for me? And in most cases, what we have to tell them is the statistics are maybe 10, 20% chance that the tumors will shrink. It's sometimes not terribly satisfying for patients. It's often not that satisfying for their doctors either. The one way, now that we understand increasingly how some of these drugs work, is to take the idea that if we know what the target is, if we know how the drugs work, can we look at the tumors and predict with a much higher degree of certainty whether a certain treatment is going to work for a specific patient. This is a whole idea of personalized medicine. So this was another example of how we sort of were able to take that concept and using a biobank, bring it to reality. The drug in this case was a drug called temozolomide. There were some early studies that we and others did showing that temozolomide was active. It seemed to be more active in patients with pancreatic endocrine tumors and carcinoid tumors, although given really small numbers, it was hard to be sure of that. So what we ended up doing is going back in the database, now looking at over 100 patients who had received temozolomide to see if that pattern really bore out. And in fact, it did. It turned out that patients with pancreatic endocrine tumors responded to temozolomide-based therapy about 30 or 40% of the time, and responses were really quite rare in patients with carcinoid tumors. So this was interesting. It doesn't really explain why that's the case, and I think as Ramesh was saying, we're really fortunate here in the Boston area in particular to have the ability to just go next door to scientists and say, well, what do you think about this? How can we understand this a little bit better? It turns out that there really is a good understanding of why temozolomide works in various different kinds of cancer. Temozolomide damages DNA, and some cancer cells have an enzyme, a protein called MGMT, that is able to repair DNA. And in cancer cells that have this particular protein, when they're exposed to temozolomide, they simply repair themselves and they keep on growing. Temozolomide doesn't work. In cells where MGMT is not present, when those cells are exposed to temozolomide, the temozolomide is able to kill off the cancer cell and the tumors shrink. So the question was, initially, can we look for MGMT in neuroendocrine tumors? And it's not really that difficult to do. Again, using the tissue microarrays from the biobank, from the database, you can see here on the left-hand side a tumor that stains positively for MGMT, and on the right, a tumor that stains negatively for MGMT. We could then go back again in the tumor specimens and see, is there really a difference in MGMT expression between pancreatic, endocrine, and carcinoid tumors? And here again, the answer was yes. It turned out that pancreatic endocrine tumors had much less MGMT than carcinoid, potentially explaining why these tumors are more sensitive to temozolomide-based therapy than our carcinoid tumors. And when we looked again, now correlating with the clinical information in the database, I think we could strengthen that particular correlation. With no responses seen in tumor cells that were MGMT positive, the response rate, though, in tumor cells that were MGMT negative was 80%. So if you think now, going initially from the clinic to the lab and now back to the clinic, this offers the possibility of having a patient come in front of you, and instead of saying, well, we have a 10 or 20% chance of responding to this treatment, we could say you have an 80 or even 90% chance of responding to a specific treatment like temozolomide, or if not, we can then go and offer them directly a different type of therapy. So those are some relatively simple, relatively straightforward ways that we can either help develop drugs or predict which drugs are going to be successful using biobanks. There is a whole nother wealth of opportunity that I just want to touch on in this particular talk, and that is really looking for new treatment targets. And here again, we have the concurrence of new targeted therapies being developed with very rapid developments in genomics. This is another project that we did a few years ago, and this was really a collaboration between Ramesh Shivdasani in the laboratory and also with Matthew Meyerson, who again we'll hear from shortly, using some new genomic technologies, at least at the time, to try to map the genomes of carcinoid tumors. This is an example of a series of carcinoid tumors, again from a database, and looking to see in those genomes, are there areas where the cancer cells have too much DNA, have too many genes or gene amplifications, or have they lost certain genes? On the top in the red, you see areas of the carcinoid genome where there's too much, and on the bottom, you see areas of the genome in blue where there is too little, showing that there really are aberrations in the genome of carcinoid tumors. The next question is, can we understand any more about potential treatment targets from these various aberrations? And in fact, just from this initial study, there were some suggestions. There was one area in particular on chromosome 14 where these carcinoid tumors seem to have a number of amplifications. Those amplifications pointed to a specific gene, and in this case, the gene was called DAD1. DAD1 stands for Defender Against Cell Death. You think about that just a little bit. If you're a cancer cell and you have a lot of protein called Defender Against Cell Death, what are you going to do? You're not going to die. And if you're a tumor, that's good. If you're a patient, that's bad. It suggests that this may be a protein that is important and may help explain why these carcinoid tumors stay alive and are so hard to treat. And in fact, a project that we're looking at right now is seeing if, in fact, carcinoid cells that have a lot of this protein are more resistant to treatments, may have a poor prognosis than those who don't. If that is the case, this may provide an opportunity as a new treatment target. We'll be hearing, I think, a lot more about that since this initial study. Genomic technology has developed even faster. I think we now have even more opportunities, potentially be able to identify even more treatment targets. Again, looking forward to the following talk from Dr. Meyerson. One other thing I want to mention is that there is yet another opportunity, yet another way that these biobanks can be used, and that is to look at risk factors for neuroendocrine tumors. We very often get the question, is there a genetic basis for neuroendocrine tumors? Are my kids at risk? Why did I get this tumor? And as far as anyone can tell right now, there really are no clear environmental factors. It does not seem to be associated with smoking, does not seem to be associated with drinking or any of those bad things. We really don't have a clear explanation for why people get carcinoid tumors. One way to try to get at this particular question is to use a study design called a case control study. And one can look at genes using this particular design. I've just tried to illustrate here on the left-hand side would represent a group of patients who do not have carcinoid tumors. And one can imagine we all have various variations in our particular genes. These control patients, or normal people, I should say, have mostly normal genes. If you then take a whole set of patients who have neuroendocrine tumors and look at their genes, you could imagine that if in fact there is a genetic predisposition, those patients may have an abnormal variation in a particular gene represented here in the red. And if we look at a lot of genes and compare the number of variants to the number of normals in cases and controls, we can perhaps dissect which genes are important and which might be contributing to the development of neuroendocrine tumors. So it's interesting. We have really just been embarking on this particular study because it does take a large number of both cases and controls. Right now, I would caution everyone that this is quite preliminary, yet nevertheless intriguing, that it turned out the gene that seemed to be most strongly associated with carcinoid and other neuroendocrine tumors is TSC2. This is a gene for tuberous sclerosis. And if you think about tuberous sclerosis, this is a rare genetic syndrome in which people get lots of benign-appearing tumors. As shown on the left, those tumors in tuberous sclerosis can arise in a number of places in the kidneys, sometimes in the brain or on the skin. And on the right-hand side, I've shown a small bowel carcinoid tumor, which also has the appearance of several. As many of you know, carcinoids arise in a series in the small intestine and often have a benign appearance. So very intriguing, I think, hypothesis at this point, still preliminary. It will take confirmation, and I want to just conclude that this is yet another reason that a consortium is important, because just looking at a single data set, you can never be sure. But if we can replicate these sorts of findings in independent data sets in much larger series of patients, one can have much greater certainty and perhaps have a much greater ability to dig down at the underlying causes of carcinoid and other neuroendocrine tumors. So I'd just like to conclude again that this large-scale analysis of biospecimens and associated clinical information can really rapidly and directly impact the treatment of neuroendocrine tumor patients. Also emphasize, though, that it's not just the collection of these specimens, but the interaction between people who are interested in carcinoid with scientists and clinicians, going from basic science all the way to clinical research to really make the most of this particular opportunity. And I think we can also see that expansion of these efforts to multiple different sites and sites that can talk to each other should also further hasten treatment advances. Thanks very much for your attention.
Thanks, Matt. We're going to hold the majority of your questions to do with Dr. Meyerson, but just a couple quick things. Are there any risks or personal benefits to a patient that decides to donate their tissue to a bank?
Just took off my mic.